Good morning, everybody. Thanks for coming in on the second day of winter. So I'm really delighted here to have a friend and colleague, Bill Murphy, talk to us today. Bill has had a long history in the immunology field. He trained at UT Southwestern, which is where his first paper was published on NK cell biology, and the really important work that we know about NK cell rejecting bone marrow allografts in mice. Bill then did some work at the NCI for 12 years approximately, leading a group in Frederick as part of the NCI. He then had a stint in Reno and then ended up in sunny California, which is where he came from yesterday. And his system was a little bit shocked as he was coming here to the University of Minnesota. So Bill's a distinguished professor, a vice chair for research, a former division director of the dermatology department. And Bill has been really very involved in recognition of immune cells to kill and attack cancer stem cells. And he really has a broad wealth of knowledge in the immunology field, working in bone marrow transplant models and graft versus host disease, and really a list that goes way too long. So without further ado, Bill, we welcome you to Minnesota. We hope to amuse you after your talk today and make sure you get to the airport on time for your 7 o'clock flight.
Okay.
So welcome.
Thank you very much. Can you guys hear me okay? Is the mic working? Okay. It is a pleasure to be here. It seems like Bruce and Jeff always invite me in winter. I don't know why. And as I was mentioning before, for some reason when I was having some meetings yesterday, my Google Maps had the Masonic Cancer Center by the Fraternity Row House, and I'm walking all along there. I don't know why. And then, so I go, Bruce, I can't find where you at. He goes, look for the red brick buildings. And I just realized he was serious. You might as well say, look for buildings with windows, because everything was a brick building. He goes, well, no, darker red. And I'm like, it's snowing out, you know? So yeah, it's hard to believe. It's a pleasure to be here. I had to say that, Bruce, I'm sorry. This is some work we've been doing for just a period of time now. When I was at NCI in intramural, one thing that always struck me was, I guess, Sam Broder said, man is a poor model for mouse cancer. It was our mouse cancer models, seemed to always work. And when it came to the clinic, that's where the problems began. And this probably dates me in the sense of, I remember giving a talk and Polly Matzinger was in the audience, and she noticed that why do people always, when their mouse tumor models, stop treating the mice after 2 weeks? All right, that's not the clinical cancer experience at all. And then it's partly because of the way mouse models are. All right, and so this was born out of an idea that we started looking at was, why do our mice seem to handle the cancer immunotherapy treatments okay, but clinically we're seeing a whole slew of things that we couldn't have predicted? And then could we make our modeling better? And so that's when I started looking at the idea of obesity. I'm going to just walk you through on how we started this, this foray. So obviously, when we're thinking of cancer and we think of how we model it preclinically, when we think about cancer, we're dealing with a heterogeneous outbred population. The problem that you see here, I had a student give me, I need a representation of what a heterogeneous population is, and I have this. The problem with this is when we see this as cancer in the clinic, a big discrepancy you can see right away if you're an oncologist is a lot of these are young. And cancer is usually a disease over the age of 55. And obviously that's something that maybe we're not modeling, especially in our preclinical models. And this is what we use, okay? So we're using inbred mice. That means they're genetically identical, homogeneous, that in the sense of also how they're raised, all right? We put them in specific pathogen-free facilities. That was a problem because if you're an immunologist, the cleaner you are, the more distorted your immune system can be. Mice don't get the pathogens we get, much less the recurrent infections. And for the most part, they're sitting and they're not doing a whole heck of a lot. And those of us that work on mouse modeling know that that's definitely affecting things because mice are living longer. Lab mice are living longer. I have a mouse that's now going on 3 years. Not that I wanted that, my students forgot to give me the inventory and I found out I've been covering for that. That mouse is probably worth about $1,000 at this stage. But that's the problem. We use young mice, right? We use 8 to 10-week-old mice because they're cheap. Homogeneous, we usually use one sex. And then we get our results. And we're usually using a tumor line that's been grown up. It's homogeneous. We inject it into a healthy mouse. And voila, that's representing human cancer progression and immune evasion. That's sarcasm, by the way. So the problem that we have is, how do we link that, right? So when you actually look at preclinical tumor modeling, other than say gem models, which are very hard and expensive to do as far as a treatment, they're great for looking at how we can see tumor regression. But if you actually had to pay the bills and say, I'm going to do treatment paradigms where I'm doing different drug dosing and you're standardizing tumor amounts and loads, it's a nightmare, and much less monitoring that. So this is a big problem as far as the demographics. The other thing that is our population's changing, right? You don't have to, you just have to see around walk around, and it's not just obesity. We're living longer, all right, older, and the obesity epidemic is growing, and it's not just in our adults, even with our kids, right? And so that's changing then our biology. It's changing our immune system. And how do we model that? And this is a problem because what we're finding now is these kinds of human variable modifying events can dramatically change outcome on almost every aspect. And yet it's not being used as incorporated like it probably should, because we want to ultimately have our in vivo veritas be somewhat veritas. That means actually mirror what we're trying to model. And so first thing I thought when I was, this is when I left NCI, was we always use young, healthy mice. They can handle a lot of our immunotherapies. The reason I was interested in this is again dating me in the era of high-dose IL-2. All right, you can give a mouse hundreds of thousands to millions of units of IL-2. It won't necessarily be happy, but it won't die. And yet I did have a colleague and friend of mine pass getting high-dose IL-2 at NIH for melanoma. All right, so there's a very big discrepancy between what we were seeing in the clinic versus what I was seeing in our models. Okay, so what we did was we got some old mice. Now, old mice here, this is 16 to 18-month-old mice. The definition of age, I know this sounds very like a philosophy, but can be really depending on what you're looking at as the system that you want to call adult, right? If you're an immunologist, what is age? All right, is it based on one thymic involution? Is it going to be based on a certain amount of immune exposure? All right, well, I did some studies and I see Mark's here. I actually wanted to look at, say, old wild mice. By the way, a feral mouse only lives about 6 months, and see if their NK cells were different. And I was having my students trap mice in Reno, and I forgot about this thing called hantavirus, so that got me in trouble. But the bottom line is, one thing we noticed is that an older, a feral mouse, their immune system is constantly challenged. Their thymus is gone much earlier versus our SPF mice. So if it doesn't have anything, you can be immunologically naive, but you are an adult, all right? And so that's something that we want to look at. And we just looked at these 2 mice and we said, what happens if we just give an immunotherapy? And one thing, bless you. So we're not SPF here, but that's okay. But one thing that we noticed right away is when I gave, this is where we gave agonist CD40 antibodies, IL-2, which we had shown previously to result in synergistic anti-tumor effects. The young mice handled it fine. But if you look at the pink, the old mice died, and they died fast. All right, that means they're completely identical. All right, bigger. All right, usually you adjust mg per kg or whatever. We didn't do that, but they died fast, and that was a shock. And what did they die of? Well, they died of cytokine release syndrome, CRS. And what we found was multi-organ pathology. We were seeing increased pro-inflammatory cytokines, and this correlated with the pathology that we observed. And it became a very a moot point then trying to treat cancer in an older mouse if they're not even going to survive the therapy. All right, so this was 2013, but then we said, well, why are they getting this? All right, so when you look at those two mice, all right, well, one thing you can see right away, other than maybe it's a little grayer, but it's bigger, right? And so when you think about what a laboratory mouse has to do all day, not much. They eat, and that's about it. And so they accumulate body fat. And so we were thinking that maybe body fat was playing a role in these exacerbated pro-inflammatory responses. So then we got some OB mice. These guys have a leptin issue, and they get morbidly obese at about 2 months, 3 months. And I see Tom's in the audience here, and I've worked with him as far as with his OBs as well. And we said, okay, what happens if we give these guys immunotherapy? And the other thing I got was from the National Institute of Aging, I got some caloric restricted aged mice, which is really kind of nice. And they're nasty, by the way, because they're always hungry. But the thing is, then we treated caloric restricted aged mice versus old mice, and then young OB versus control. And what we found was, first and foremost, when we had the caloric restricted aged mice, that's the dotted line, they survived, okay? Whereas the aged controls died. So clearly body fat is playing a role in that meta-inflammatory state, that pro-inflammatory cytokine storm. When we looked at the obese mice, we found they still succumbed, but there was a delay. All right, and then this is obviously morbidly obese. All right, when we looked at diet-induced obese mice, these are mice that you make fat. They also would have a delayed, but they did have a pro-inflammatory storm, but it wasn't as profound as either the aged mice. So clearly, age and obesity all can contribute to the cytokine storm. And what we found is if we could actually block certain things, certain pathways, and the most notable one was if we remove macrophages, the mice now could survive. So it was a dysregulation of macrophages, and if we block TNF, we could also protect. So this was actually building off the bone marrow transplant GVHD, where the cytokine storm actually originated as far as a concept, other than also in sepsis. But clearly immunotherapy, immunostimulatory therapies can exert a similar kind of paradigm. And obviously in cancer, that could be a problem. And those of us that work on CAR T cells can tell you that is a problem, as well as also like with COVID. So the bottom line is we found, yeah, obesity plays a role, and an important role. And actually it could be very important then to mirror and incorporate in our therapeutic interventions, because bottom line is you want to be able to then figure out, like right now they're using IL-6 blockade and TNF blockade, both for CRS and CARs, but also looking in COVID, that we can understand. And that we're saying this is mainly with immunostimulatory therapies, okay? And we can see the same thing if we, and I don't have time to go into it, if we infect mice with an acute viral infection. We see the same massive pro-inflammatory cytokine storm. And then for our papers, they were saying, okay, but what about if you gave CTLA-4 or PD-1? Okay, what about, does that also do it? Okay, that's a good point. So we said, okay, we'll look at that with the idea that maybe we'll see more toxicity, right? That's the idea. Obesity, bad. All right, we'll see more toxicity. So what we did was we made DIO mice, that's diet-induced obese mice, that are placed on a diet, 60% high fat, which is lard, for 3 months. They get a nice weight differential. They don't have the metabolic effects at that time. And we gave tumor, and we just gave PD-1 monotherapy. Now, for those of us that do preclinical modeling, PD checkpoint monotherapy as a rule doesn't work very well in mice. In people it does. But even Jim Allison showed with CTLA-4, he had to give it GM-CSF to really get the effect, mainly because when you think about it, mouse tumor lines grow really fast. All right, the mouse usually will live, give the tumor, in 3 weeks later, it's done. And so that's an issue where maybe that's why it doesn't work. It's been shown monotherapy with PD-1, you have to give it with radiation or CPGs or something like that. We just did monotherapy. And what we found, we noticed right away, the red, tumors grow faster, surprisingly, in fat mice. Okay, now that's been shown by others. Probably it can be due to a plethora of reasons. That's the problem when you do obesity studies. Everybody wants the one thing, the one fatty acid or whatever. It's probably, yes, you're in a nutrient-rich environment, there's immunosuppression. Overall, it's been shown even with clinical outcomes, obesity is a poor prognostic indicator for cancer, and it grows faster. But what we found is if you look at the pink, if we gave anti-PD-1, we saw anti-tumor effects, but we didn't see it in our lean mice. Okay, so this was surprising. And presenting this as a waterfall plot, we only saw the anti-tumor effects and the therapeutic efficacy of checkpoint in our obese. All right, and that's what starts the obesity paradox. About the same time we were doing this, McQuaid at MD Anderson, using clinical data, said, my obese patients do better. All right, and not just with checkpoint. The clinical outcomes in obesity were actually better with molecular targeting agents, even with chemo. And now we're seeing it in our mouse. We actually then looked at, this is with Oklahoma and now Roswell Park, clinical outcomes looking at a variety of tumors with checkpoint. High BMI, that means over 30. That's what we use as a cutoff for obesity. And BMI, by the way, sucks as the only way to determine obesity. There's a lot more to it. Muscle mass is now being more appreciated. Sarcopenia, you could have normal BMI, but that's also a poor outcome. And it's not telling you anything about metabolically what's going on, but just simple stratification of greater than 30, less than 30. We could see a pretty clear demarcation where there was a survival benefit with checkpoint block. So what this was telling us is as opposed to immunostimulatory therapy, checkpoint, actually obesity is a good thing. And that's not to encourage people to go to McDonald's and everything, but it was telling us that it's not as simple as one or the other. So we see that clearly, this is what's interesting. The tumors grow faster in obesity. So you're dealing with a race between the tumor and the immune system. But in the case of checkpoint, it looks like by taking the brake off, the immune system not only catches up, but can overtake. And this also then correlates with the outcome. It's also interesting too, that even though we're looking at this kind of, and this was all mainly due to T cells, CD8 T cells, obesity is very complicated. It depends on the type of tumor, depends on really the milieu involved. And it's clearly that while there can be protective effects, there's clearly something that we're going to have to understand more, how we're going to apply this. As far as, is it body fat? Is it the diet? Is it sort of other things? And then also other immune cell types, because I had to do the handout to call out for NK cells. NK cells are paralyzed in obesity, all right? And so maybe it depends on really what type of tumor and how sensitive it is to a particular immune cell type. But these are things that need to be assessed. But are there sex differences? And the McQuaid study in Lancet Oncology indicated it was. In her study, she showed it was only high BMI males that actually had the beneficial effect. High BMI females did not, all right? And our Nature Med paper came out about the same time, and we were showing basically a nice effect of obesity in our mice. But one thing that I forgot to do was, it's NIH mandated, but I only use male mice. Why? Because you can only buy male DIO mice, all right, to purchase. Otherwise, you have to generate them yourselves. And so it was just easier for us. So all our studies were male mice. So then we said, okay, if this is what the clinical paradigm is showing, let's look at male and female, make them both fat and see what happens. And so the McQuaid study said it was, males did better in the melanoma. There was also a recent paper in JCI. Estrogen receptor seems to play a role in M1, M2 polarization in macrophages. However, there's also reports that androgens are bad on T cells. So you've got sort of like, all right, good, bad, what are we going to do here? So the bottom line is, let's look at this. And so we wanted to see, is this paradox exist equally? And when we looked at our clinical data and we then re-stratified on male-female differences, we saw what McQuaid saw, and it was only the high BMI males that exhibited the beneficial effects of checkpoint block. All right, the high BMI females did not. Now, this is obviously tough because you're going to, in a clinical correlative study, depends on the cancer. Some cancers, if it's over the age of 55, we're also including, postmenopausal. There's going to be a lot of variables going on there, but at least this gives us something that we are seeing that same trend. So then what about in our DIO mice? So we basically made our male and female mice fat. You put them on the high fat diet. And what you can see right there is the red is the male, the blue is the female. As far as, the red squares are the males on the diet, and then the female on the diet is the red circles. Males put on weight faster, okay, which is kind of not good if you're a male, and I can probably attest I feel that way sometimes, but the bottom line is the females catch up. All right, so that's good, all right, because one of the problems when you're doing obesity studies is you want to level the playing field and you want to have comparable weights. But that's what we went with. And then we basically said, all right, let's look at the same kind of treatment paradigm, male and female, and see if we see what we see in the clinic. And sure enough, we do. The male mice responded to checkpoint, the females did not. Okay, so we are being able to recapitulate somewhat what we see in the clinic as far as a differential sex-linked difference between male and females with regard to anti-tumor effects. Well, okay, let's nail it. Let's say, are hormones that important? So we did the same study, only this time we overrectomized our female mice and then put them on diet. And so first thing we noticed is that the overrectomized females gain weight as quick as the males. Those are the two almost superimposable top growth curves of the male on the high-fat diet and the overrectomized female on the high-fat diet. So we are actually seeing that by taking away that aspect, we're already seeing metabolically a big difference in body fat deposition. And then when we looked at anti-tumor effects, we saw now with the overrectomized females, we can see a beneficial effect in this waterfall plot in a reduction in tumor growth, similar to what we would see with the male mice, all right, following checkpoint block. That is, simply by abrogating that estrogen response, we're now able, in a high-fat diet scenario, to get a beneficial effect. So that's actually encouraging, suggesting that what we can see is there are sex-specific differences. Those of us that work with grant reviews and deal with NIH grants, I remember when they first mandated you had to do male and female, all right? And I was like, nobody does that, by the way. It's so expensive to actually do male and female. And males fight. I mean, usually we use female mice. And that's the other thing. What's interesting about DIO mice is they're placid. The male mice don't fight anymore, all right? Normally when you do male mice, they fight like crazy. You get them fat, they don't do anything. They're just really happy campers, which is kind of an interesting aspect of society I think we need to look at. But anyway, clearly there are sex-specific differences, and I hate to say it, NIH is right. We should be doing these kinds of studies in our preclinical monitoring because you would miss it otherwise, or you would get the wrong idea on what is going to work or not work, because that's not reflecting your whole population. The other thing that's important is this is just most of us that do preclinical modeling only work on 2 strains of mice. We do B6 and BALB/C. That's it. Unless you're doing some crosses and you're doing SJL, for the most part, all the tumor lines that we use are in those 2 strains. All right. And those of us that know mouse immunogenetics, that's like saying you're looking at one person. And that's like my response now, if I'm a B6, is representing everybody. And that's just not the way it is. Obviously, there's going to be genetic components we can't fix, but also, as done by classic work here, maybe making the mice more human-like with their immune exposure could be important. But anyway, there are sex differences, and it's kind of nice that both preclinically and clinically, we can see a similar trend. It's not like it's totally out there. We are clearly seeing these sex differences have different T cell exhaustion signatures, which I don't have time to go into, as well as the M1, M2 polarization. The fact that ovarectomy in the DIO female that could recapitulate the male actually gives you the idea that maybe we can be using hormone blockade, tamoxifen, all right, in a high BMI scenario to also augment. The other thing to keep in mind is, you know what they say about PhDs, I know a lot about very little. So I know a lot about immunology, but I'm really terrible about physiology. And I had to learn about metabolism and how males and females also put on body fat in different areas, subcutaneous versus visceral. It's a real problem then figuring out brown, white fat. There's a lot of issues. Even how we keep our mice. All right, most Betsy Rapaski and others are saying most of our vivariums are way too cold for a mouse, and so there's an artificial effect of that. So these are all things that I think people view the mouse as a test tube, but really that's supposed to be the in vivo model that we're representing as far as in our patient population. And also being at NCI and doing this for so long, we're very averse to adversity as far as negative events. Nobody wants to see this causes toxicity. Nobody wants to see this doesn't work and causes toxicity. So we always see the model such that you make it so it does work. Even if you're giving, I've seen models where they actually treat with the cancer therapy, then they give the tumor, which my head explodes. So the bottom line is that's going to be an issue that we need to figure out short-term gain, long-term loss, as far as maybe it's better to push things so you do see these kinds of effects, so you're not surprised as when they occur. Then I've been working on bone marrow transplant, and that's why it dates me because I say bone marrow transplant for a long time. So we said, what about in BMT? So preaching to the choir here, we all know that bone marrow transplant is for a variety of diseases, but it's really also with the advent of CARs that we're seeing now applications. In my institute, we're doing CARs for sickle cell and HIV. So you're seeing bone marrow, and you're always involving a Cy/Flu or a cytoreductive conditioning. So to get the T cell engraftment, you're immunosuppressing. And if you want to do a third party, you're really hitting because you want those things. So the bone marrow transplant field is actually giving us a lot of lessons on how we're going to apply cell therapy. All right. And it's not just in the context of letting the bone marrow transplant do everything. You're using that to create the environment so the cell transfer that you do works, but some of the same toxicities and effects you may still see. And working in bone marrow transplant, obviously allo transplants, you get graft versus host disease. All right. These are the classic studies by Sprint, even with Metawar, showing that MHC disparity and immunosuppressed environment, you have now a bad scenario. And so the question is, unfortunately, that bad scenario also gives you a good scenario. E. Donald Thomas with the Nobel Prize in bone marrow transplant, you get a graft versus tumor effect. All right. The first evidence I found that immunotherapy can work is that you actually get a GVT effect in a bone marrow transplant. So that's good. Problem is, as those of us that work on it, you're walking a tightrope between graft versus host and graft versus tumor, and that's been the holy grail, separating the two, which I'm personally not sure you can. But the bottom line is, this is where cytokine storm originated, as far as they noticed that once GVHD is ongoing, you get a cytokine storm that then it needs to be, and that's where a lot of the interventions, IL-6 block, TNF block, were looked at. All right. And increasing role of the microbiome. I didn't mention the microbiome before. Microbiome is very important, right? We appreciate that now. Well, what does obesity do? It really changes the microbiome. So what's the effect? So not too surprisingly, we made fat and lean mice, gave them a bone marrow transplant and do an allo. And what we can see, the red is the fat mouse recipient, the blue is the lean. Not surprisingly, you get a cytokine storm exacerbated when you use obese recipients. But what I found was fascinating was this was a standard combination of allo transplant where they get an acute gut graft versus host disease. But we actually use strain combinations where normally there's a chronic GVH that occurs later. It's a minor MHC mismatch. You only get a skin manifestation, and it's not lethal. We made those mice fat, and they all died from acute gut GVHD. We actually changed almost the phenotype of a strain based only making it fat, which is fascinating. And this is all mediated by CD4 T cells. So we were seeing the CD4 T cell response was exacerbated. And again, we saw the cytokine storm. And similar to the immunostimulatory therapies, cytokines were up, and we saw a whole bunch of, and this is mainly in the gut, which is also interesting. GVHD can attack multiple organs, but the gut was really hammered. And so working with Bruce Blazer, who's in the audience, hi Bruce, Shernan and others, and then also with Jamie at Mount Sinai, they also were looking at outcomes, allo, and could see a similar paradigm. Now, checkpoint monotherapy is easier to stratify because the conditions are pretty much comparable wherever you're going on how it's being given and used. Bone marrow transplants, those of us that work in that area, it's a nightmare looking at different places because of the conditioning and all the different other variables. But you did see a similar thing where, as opposed to checkpoint, high BMI in this situation was a negative correlated outcome. So again, mirroring what we would see in our other models, the mouse and the human scenarios can be linked. But you have to have at least some of the human variables like obesity in consideration. And what was interesting for us is this wasn't just an immune reaction. When we looked in the guts of our obese mice, we saw increased gut permeability. We saw decreased microbiome diversity. We saw increased radiosensitivity as far as endotoxin translocation following radiation. So obesity is basically further amplifying the inflammatory environment even before the immune cells get in there. And so that's something that needs to be taken into consideration. I don't have time to go into it, but in our paper in STM, we showed the decreased microbiome diversity existed both in high BMI patients and our DIO mice. And if we gave prophylactic antibiotics, we could partially, we didn't cure, we could partially counteract the increased pro-inflammatory storm. Also, we're not seeing a male-female difference now, okay? And that's important. So in other words, everything can be viewed as strength of signal in the sense of if the signal is very strong, where it's like a bull in the china shop, so to speak, male-female differences we didn't see. The female DIO mice succumb just as fast as the males. Now, granted, these are very extreme models where maybe if we fine-tune enough, there might be a difference, but at least versus say checkpoint. But the bottom line is we did not see a difference here. So then we said, okay, we had shown earlier that if we block TNF, all right, we could at least partially protect the aged mice from the CRS. And obviously, Carl June and others have shown with IL-6 block, you can block, help the CRS with CARs. And so, those of us that are immunologists, one thing that's always struck me is everybody tends to focus on one molecule or one pathway. And so we'll see TNF block by itself, IL-6 block by itself, but I couldn't find anything in the literature where let's give them both together. Because if you may look in the, you saw both going up and they're both pro-inflammatory. Ergo, there may be some compensation that, just like with cancer chemotherapy drugs, just doing one drug usually is not optimal. If you give multiple, even if they do similar mechanism of action, you may get a more beneficial effect. So we actually tried giving IL-6 and TNF blockade in our DIO and mice. And sure enough, when we do the transplants, you can see in the green and the blue, that's the single blockade. But when we give the double blockade, that's on the top, we saw much better protection from GVHD. So what that's telling us is that when we're looking at some of these regimens, just like with cancer treatments, where you don't just give an immunotherapy. Usually you're giving it in conjunction with conventional therapies as well. That means maybe with immune interventions, we should be thinking about giving some of these approaches. People have looked at depleting macrophages, CSF1 receptor blockade, and it's been equivocal results. But sometimes I think we ask too much of our therapeutic regimen to take care of all of the aspects of that pathway or circuit by just targeting that. Whereas if we use multiple pronged approach, sort of like with, say, heart therapy and HIV, right? You give multiple antiretrovirals, maybe we do multiple anti-inflammatory, it allows us to get a much greater penetrance such that we see greater protection. Importantly, too, you have to do the obligatory, what about anti-tumor effects? And so doing the same double blockade, we gave basically these mice, the fat mice, tumors, and then we gave them bone marrow transplant. And then we imaged the tumors. Now, if you just do a bone marrow transplant, and that's the middle group there that you see with the A20, which is a lymphoma, you see those, they're not dying of tumor, they're dying of GVHD. So what's happening is, yeah, you look and the ones that survived, the tumor's gone, but unfortunately we lost half the mice due to graft versus host disease. So that's the caveat. If you look at the ones that got the same transplant, but now you add the IL-6 and a TNF block, we see much greater survival, obviously, without the GVHD. Now you could say, well, wait a minute, TNF blocks associated with, say, breast cancer and infectious disease susceptibility. It really may depend on what you want to do, not a prolonged, but if you can use it at a surgical kind of a component, you might be able to have all the benefits without the loss of immune effects later on. Because IL-6 and TNF also can be great, and especially IL-6, tumor promoting agents, promote tumor progression, and myeloid and MDSCs and everything else. So there could be even added benefits, and that's something that needs to be looked at in our obese situation. I can tell you when you put a melanoma on a fat mouse versus a lean mouse, the tumor looks different, and I'm not a pathologist. I mean, it's got a much leakier vasculature. You get a much more inflammatory component as far as that. So there could be other aspects of blocking some of these pathways outside of just helping the T cells work better that we could exploit. So our conclusions is that, you know, basically we see the same thing. Obesity can be bad if there's systemic immunostimulation. But if we block certain pathways, we can possibly use this to our advantage because you can get the immune potentiation without necessarily the toxicities. And this may actually have applications with CAR T cells, may have applications with COVID and other kinds of acute inflammatory conditions like acute viral infections that could be looked at. So finally, this is the fun part. I have enough time, I think, is I wanted to say, okay, everybody asks, do you have to be fat? Okay, and maybe the fat's not PC, but do you have to have increased adipose tissue content? Or is it the diet? Okay, so our mice are on a high-fat diet. We don't use a Western diet. A Western diet is high-fat, high-sugar, because frankly, that's a nightmare metabolically on our mouse studies. All right, and the other thing is, what about the microbiome? All right, what's playing the dominant role? In a GVHD at least. Now you're seeing a lot of microbiome work. Everybody wants their secret sauce, the microbiome that is only good and correlates. And you're seeing it even with checkpoint. Tom Gajewski, Lauren Zivogel have papers, and you're seeing a lot in bone marrow transplant. But in our mouse models, and I think that's what people don't appreciate with your preclinical models, you can actually do mechanistic studies. You can actually tear it up, where otherwise you're doing correlations with clinical data that gives us kind of an advantage. And this is where if you understand mice, it's actually kind of fascinating. You put mice, B6 mice on a high-fat diet, they all get fat, okay? You put a BALB/C strain mouse on a high-fat diet, only 50% get fat, females, all right? The males all get fat, but the females in a cage, 3 will get fat, 3 won't. Well, actually can't do that. We do 2 and 2. We have 4. I'm sorry. I don't want to get in trouble. Okay. No, we do 4 per case, so 2 and 2, something like that. But that's fascinating. Think about that. These are genetically identical, okay? Housed completely the same, and yet have a totally different phenotype in a cage, all right, in response to just diet. And they're all eating. Now, those of us that work with mice know there's a lot of social hierarchy that can go on. But those of us that work with mice know it's mainly actually in the males. They're the ones that are a nightmare sometimes, putting them together, especially if you're doing mixing studies. Females don't show overt aggression to that extent. And yet it's the females that show a differential. And so I think that was but that gives us an opportunity. The opportunity, that means we have mice that actually have body fat, and we have mice on the same diet that don't have body fat put on. So that we can address the diet issue versus the body fat issue. And so sure enough, if you look at the upper left, if we look at the weight gain after 3 to 4 months on diets, and our female biopsies on the high-fat diet, you can see a clear demarcation, okay? And what's fascinating, this is, you gotta love science. What's fascinating is if you separate them out, okay, and then keep them on the diet, they keep that phenotype. So they don't so it's not a cage hierarchy. Then we did something that was really neat, and I had to get IACUC exemption for this. So we single-housed female mice, all right? Because we're thinking maybe it's something, one's intimidating the other, it's a stress thing. And then I'm saying, well, is the dominant the one that puts on the weight or doesn't put on the weight? Those are kind of things. But I single-housed female mice, and the same 50% pattern emerged. That means in the absence of others, something is set. It must be, I'm thinking it's birth order or something like that. Go figure. I think it's real. But even after 4 months, we still see a 50% with our female mice, which I think is pretty cool. And then what we wanted to do was, okay, first thing, if we look at our high-fat diet but lean female mice versus our high-fat diet fat mice, and we looked at GVHD, again, we saw an in-between result. You gotta love it, all right? So the bottom line is the red triangles are our fat female mice that on the diet, and the orange squares are resistant female mice that are on the diet, but not putting on the weight. In fact, their weight was not different statistically from the control diet mice, but we still saw a partial effect on GVHD outcome. And we could correlate, if we just bled the mice for serum cytokines, we could see the ones that succumbed, obviously, this is a keen grasp of the obvious, had higher cytokine, all right? Whereas the ones that survived had lower. So, you know, for those of us that think an inbred mouse, by being completely identical, all right, genetically, we've controlled for all these variables. We're still seeing tremendous heterogeneity here, only with one variable that we should be seeing. You know, you can imagine then extrapolating this to an even more with outbred, looking at different exposures to different things, looking at age. Everything's the same here. That's amazing to me that even within a cohort where everything's the same, we still see a difference as far as outcome, which is life or death in this case. And when we looked at the microbiome, we found that the diet does matter. Okay, that both the susceptible and resistant mice had similar restricted diversity. Okay, now those of us that also do mouse studies know mouse microbiome studies are a real pain because mice are, how do I say it, cacophonic? They eat each other's poop. So they'll normalize within a cage. Okay, but what was interesting, then you got to ask yourself, what about the single house guys, right? That's what was, so the bottom line is the fact that the susceptible resistance showed decreased diversity means decreased diversity wasn't alone enough because clearly the body fat gainers had a much worse outcome. So what's the problem? Well, because they had comparable why, we still don't know. It may be stress. And as I mentioned before, when we single house females, we still put them on the high fat diet, they didn't have uniform gain. Well, what about the microbiome? And what was fascinating is the single house mice, the single house female mice, all right, also all had restricted microbiome, even if they weren't on diet, okay? And one thing out of scouring the literature, I found that, well, because you know, if you work with mice, single housing is considered a stressor. That's why you have to get an exemption to even have that happen unless the mice are fighting. And well, how do you measure stress? Well, you can do corticosteroid measurements. That's really a mess, okay? Actually, the thymus is very sensitive to stress, but that, you know, you're not gonna knock the mice out to see the thymus reduction. The microbiome is very sensitive to stress. There are actually papers on that, such that the moment you single house any mouse, you get restricted diversity in the microbiome, okay? Well, that's pretty cool too, because now we have mice that are on regular diet, not fat, but have a restricted microbiome diversity comparable to the other guys. What about their GVHD? So I'm just going to summarize this as a table. And what we found was in our original paper, that was like 2 years ago, we said obesity causes a pro-inflammatory storm and microbiome alterations. And this is the mechanism. Well, guess what? I was wrong. Okay. Is this recorded? No, I was partially wrong. So the thing is, yes, if you look at this is looking at outcomes, acute GVHD outcomes. If you look at the group house mice, the DIO susceptible, that means they have body fat and high fat diet. They have the worst outcome. The DIO resistant, they're on the high fat diet, but not gaining weight. They have a partial protection. Okay. And then if you look at the microbiome diversity, that's what we started with when we wrote our paper before on the outcomes. It looked pretty clear. Microbiome and antibiotics did have a protective effect. So it wasn't, you could see where we can make that kind of a rational conclusion. But the single house guys, well, they had the same GVHD patterns that you can see on the right as the group housed. If they were DIO susceptible and they put on the body fat, they had the worst outcome. If they had resistant, they had the same, it was exact. But the microbiome diversity was restricted in all of them. So what this was telling us is that it's not as simple as this, or this. It means that obesity, body fat deposition, ultimately is the major driver, all right, for a negative outcome. Other things can be what we call amplifiers. So there's a difference between being an inducer versus an amplifier. So obesity clearly is an inducer for an acute GVHD because we were seeing it in strain combinations that didn't even have it. That being said, diet also seems to play a role. And that's as important too, because what this means is, now this is a negative outcome, but the question is then if for checkpoint, maybe we could be putting mice on diet, they don't put on the body weight, and they still have the beneficial immune effects. That's what we're looking at. It also means that the microbiome diversity itself is not going to be the home run in this case, but it does play a role, and it's something that needs to be considered, okay? And so from all this, I think you can gather, A, there are sex-linked differences in immunotherapy outcome, and human modifying factors like obesity are really critical to assess. If we were just doing all of these studies, which say 10 years ago, I did all these studies in just young, healthy mice, you'd miss what's going on as far as now that we're re-stratifying clinical data on BMI, which by the way is surprisingly hard to do in this era of personalized medicine, that just getting BMI is like pulling teeth sometimes. It's amazing. I was telling at UC Davis, we should be doing that 10 years ago, we should be stratifying. And you would think I'm saying we need to walk on the moon. But the bottom line is that's amazing that we're not even looking at even more sophisticated measurements of metabolism in our patient population. And we're starting to look at like imaging for body fat deposition, muscle mass, things like that. The mouse modeling isn't even there, right? And we should be there. With GVHD, clearly these can be negative correlative factors. I thought what was interesting was the impact of stress. So stress that's not observable, all right? So I'm on the UC Davis hierarchy, and how do you know when a mouse is in pain? How do you know when any animal's in pain? It's very difficult. How do you know when an animal's stressed? It can be very subjective. The microbiome seems to be a pretty good indicator, but then you got to wonder about your cancer patients, because mice don't know they have cancer when we inject our tumor lines, all right? And so you got to think about how that is going to potentially affect not only the immune system, but also even the microbiome. But I think this could be very important if we want to dissect also GVT, anti-tumor effects, versus the negative inflammatory effects. So here's the obligatory cartoon at the end. So you get, is obesity good or bad? And the answer is yes. But it's really, when you think about it, I always think as an immunologist, the concept of checkpoint block, the reason that was so paradigm shifting in the sense as an immunologist, when I was at NCI, we used to call it the National Cytokine Institute because we gave high dose IL-2, IL-12, whatever, even TNF, things like that. And cytokines aren't hormones, and people would get very sick and they would die. But you're putting gas, you're giving supraphysiologic amounts, all right? The concept of checkpoint is you're taking the brake off. It's sort of the analogies with oncogenes and tumor suppressors. It's hundreds of oncogenes, dozens of suppressors. That's the rule. Same thing, the brakes are more important than the turn on signals. And the tumor knows that, because the tumor is not trying to deprive the immune cells of their oxygen. It's trying to turn them off. And the immune system, in my mind, is all about control and turning off. And that's what made this a totally different outcome. So in this case, this could be LPS, sepsis, BMT, systemic immunostimulation, not good. All right, you get the cytokine storm, you get macrophage dysregulation, and then GVHD. But also adding to this is the concept with aging. All right, if I break a leg right now, it'll take me months, if ever, to heal. My kid can bounce off in about 3 months. All right, we know that even with chemo, when you're younger, you repair more, and as you get older, things break down. And that means it's not just more cytokines, it's the receptors, it's on our tissues. It's also the damage that we've accumulated and all that. That's creating more of the environment, that cascade that we're talking about. However, now with the idea of just taking the brakes off in a gasoline-rich environment, okay? Because remember, in cancer, it's a fight for resources. The tumor's winning, all right? And so that's why cachexia is a real problem when you're thinking about cancer, all right? And the immune system's gonna lose that. Bless you. Here, we get the opposite, all right? There are sex-linked differences, but we may be able to exploit that. We need to understand that more. And it may depend on the tumor type. It may be M1, M2 polarization, it may be T-cell exhaustion, it may be a slew of other things, but it needs to be looked at. I tell you too, there is a reduced effect of cachexia. Usually in IACUC, you have a 20% weight loss cutoff to have to euthanize the animal. Believe it or not, you can get an exemption for DIO mice. They can live up to 50%. Because they're obese and they're fine. And so that means they can actually handle the stressors a bit better. And that's something to keep in mind. And we saw increased T-cell responses with regard to checkpoint response. So the conclusion from all this is, I think we need to get beyond the 1970s and 80s on how we do preclinical modeling. All right, I think people are, personally, this is my band, I'm on a pulpit right now, but we're going into the humanized mice, PDXs and all that, but that's even more far removed because then you're really not incorporating these things. But I think that you need to be looking at these modifying age, obesity, sex, prior infection exposure, things like that, and see how that affects the immune system. And in our tumor models where there's really immune evasion going on. Because a tumor that's grown up in vitro under confluent conditions, there's no immune pressure on that. And you're putting it in, the tumor is just trying to engraft. It's not trying to evade the immune system, all right, even in an immune deficient animal. And so that presents a problem then, saying that I personally feel the checkpoint responses we're seeing here in our tumor models, mouse models, is nothing like what we're seeing in the human scenario, where epitope spreading is occurring and it's more prolonged. People are on checkpoint for years now, right? The most we can give it to a mouse is usually 2 weeks because we're not using mouse-sized reagents. We're using our hamster or a rat anti-mouse. You get neutralizing antibodies after about 2 weeks in a normal mouse. So you're not even recapitulating the paradigm, much less combining it with other therapies. These are the people in my lab. I just want to also acknowledge long-standing collaboration with Bruce. Bruce and I have known each other, what is it, 35 years. I first met him as a graduate student and he was a full professor. No, I'm just kidding. But no, it's been a long-standing and it's one of my best friends in science. Jeff and I have been working together and it's been so great talking to somebody that actually knows what NK means other than, and then also just being a leader in that area. My connections with, and Tom's here too, with Minnesota's been fantastic if it just wasn't so darn cold. But then again, I suppose it last 2 months ago it was 116 degrees in Sacramento. So I guess it's depending where you want to go. But anyway, these are the people in the lab, and I'm happy to take some questions.
I've got microphones for folks with questions. Jeff?
Hey, Bill, that was really great. I just have one question. Do you know anything about, the duration or the reversibility of these phenotypes? I mean, I'm always trying to think of actionable areas to impact on a clinical outcome. So is it well studied? Do you know anything that's worth commenting on?
Yeah, I mean, there's been studies looking at patients with bariatric surgery. NK cells stay paralyzed, apparently. I think the problem is when you're dealing with clinical studies, it's almost impossible with all the different variables coming into play. Mouse studies are even more of a nightmare because it takes 3 to 4 months just to make mice fat, much less than to put them on a diet. And then you have the age variable that goes in. That's been the big issue for me, is I had to redo all my control studies because my mice are 7 to 8 months when we're starting. All other tumor studies are usually 8 weeks old. So even mine. So you almost have to rederive on a new baseline that you're then comparing to. And so nobody's really done that at all. And it's very expensive, right? So like I said, our aged mice during COVID, we had to shut down. And I had mice that were 24, 26 months. And we're going to infect them with virus as a way to stay. And they were like $600 a mouse otherwise. And that's hard to maintain. And NIA is cutting back on their colony, right? And that's a huge problem. And so I think the NIH doesn't appreciate the necessity of these kinds of studies because they're very expensive and take time. And most grants are not geared that way.
Hi, Ronnie. I just want to build off of Jeff's question in that, have you looked at the immune cells in the obese or aged mice at an epigenetic level? So like, how malleable are they, or once they are,
Yeah, I think they're very malleable because when you're dealing with mouse age kinetics, 1 week is for a mouse, when you're age equivalent in human, you're dealing with, when you add that in time, it can be considerable. The problem is I find, and maybe Mark and if David's in the audience, he knows, is the problem with SPF animals. So, and then the role of the thymus. So a mouse thymus is not the human thymus as far as, the mouse thymus can keep going. The memory naive ratio, and even an older SPF is nothing like in the human. And so I think that's a big issue because the malleability depends on what your, how many are naive, what's set memory, what's your antigen exposures and things. So we need to be maybe infecting mice with MCMV and let them go, so we can actually sort of mirror some of this, because I think it can give an artifact. It's like looking at a kid's ability to respond and reverse versus an adult, right? It's a lot easier. You see much better effects the younger you are in the reversal and I think it's going to be difficult to really model that in a SPF mouse.
That was great. Thank you. I have two questions. First is about the immune blockade. I think recently there are data showing that PD-L1 can actually target senescent cells and improve aging phenotypes. So I'm wondering, in your first part, when you treat with immunotherapy in mice, does that improve the metabolic phenotypes of these mice? Is that associated with your outcome?
Yeah. So, and again, it's sort of related to that. The problem when you're doing metabolic components, it's dynamic, right? And so our DIO mice don't become insulin resistant if you're doing a high fat only versus Western diet. We don't get insulin resistance until like 10 months, 12 months. And so those are almost different kinds of paradigms. And then what your readout is, right? So what are you calling senescence and what are you looking for? Because we can see evidence in the literature that NK cells actually increase neurologic dysfunction by attacking senescent cells and blocking them. And I'm also seeing where NK cells may be good by inhibiting responses. So I'm not very hopeful that it's going to be as simple as the immune component targeting one pathway, especially there's a lot of emphasis on PD-1, PD-L1. Obviously, we know there's TIM, LAG, TIGIT. I mean, there's a whole bunch that make it, there's compensatory things that can be going on. So I haven't seen that yet as far as, and there may be waves as far as you may see an initial effect, but then you may see a normalization. Anytime you try to block something, it used to be, I joke with my students, if you make a knockout and you see an effect, that's great. But if you make a knockout and you don't see effect, that means it's even more important because there's something compensating for it, right?
Yeah.
They don't like that because then they got negative weight. But I think that's going to be the issue. You may see something and then it's the window and what your readout is.
Yeah. My second question is about the gut. So for intestinal epithelial cells, they have these absorptive and secretory lineages. When you challenge mice with high-fat diet, they actually remodel and then make more of these absorptive lineage to take up the fat at the expense of these secretory lineages at, for example, tough cells or goblet cells. Have you looked at those, cells in your models?
No,
and does that contribute to?
Yeah, we actually, and that's another thing that being a bone marrow transplant, you're sort of a jack of all trades, master of none. Because my knowledge on the gut is, we're seeing more endotoxin translocation, more permeability. But I think that's important. And I'm actually kind of frankly surprised the GI world, the Crohn's disease and people we're not using these models more to actually address some of those, because the problem again with bone marrow transplant is you're giving cytoreductive, you're giving lethal radiation. So you've got the radiation damage effect too. I think that, and it's actually interesting too, you raised a good point. I actually never thought about that. Increased gut permeability and endotoxin translocation may actually be a good thing for checkpoint. I know, and that never occurred to me that actually, because as we know, what happens locally doesn't stay local. You can see then systemic immune effects. And that may actually be one of the reasons why high BMI do better, is because of those effects at checkpoint. Actually, that's why you give talks, because that's a good question.
Okay, we've just got our last question down here.
Quick question. Quick question, Bill. Have you tried transferring T cells from obese mice or H mice into normal mice and evaluate? I know it's a hard experiment to do.
No, it's not hard. It's just hard to interpret. I mean, these are the old, God, remember Bruce Rongress, the old studies, the old into the young bone marrow and things like that. We have, and I would like to say it gives us clean data, but it doesn't. And so in that sense, I think part of it's our limitations of what we know about the cells we're transferring and what's considered the conditions to allow them to engraft. And I think that's a huge problem. And so I think the problem I'm finding nowadays is people can see what they want to see, and you can gear any model for that. But I'm not sure those really get at the answer on environment versus, I think that's an important question. I just think, unfortunately, the modeling system, whenever I see adoptive transfers, I get a little nervous on, I'd be careful that you've really nailed one thing, because it's like in a vacuum. It's and it's the uncertainty principle all over again. You just don't know how much you've distorted, and is that really what's going on? But I think those are good questions.
All right, great. Can we say thank you one more time for Dr. Murphy? Thank you.